besides the point. Anyway, so where we had left off with last time, we're going to take this information and we will replicate that. That's what we're going to be talking about today, but from prior knowledge, okay? If you don't know what these base pairings should be, okay, just continually practice that. So you tell me what's going along the right-hand side here. And some of you might not be able to see it. A and A. Oh, yep, you're right. These aren't quite straight across from one another. Okay. Yeah, that's. I was seeing if you were paying attention. Okay. And then, okay, so another G. Now, like I said, they're not quite straight across from one another. There we go. Okay. So now what we want to put in between here is some of these guys. This will make more sense what these dotted lines are. Now, whether you choose to draw this, that's entirely up to yourself, but I would suggest that you do. If you didn't get it down, then maybe head back to the science channel, and you're more than welcome, of course, to view that. Okay. Is Amanda the only one gone? 5, 10, 14, okay. 16. All right. Okay. Just a little bit of a background here. Now, this is something that the author probably does not have as far as information is. Okay. When you look at these two individuals, okay, James Watson and Earl Crick, discovering this landmark, well, having this landmark discovery, probably gave them this prize in either biology, or if you want to call it genetics, or what have you. What is the top prize to earn in whether it's science or literature? Say that again. The Nobel Prize, whether I believe it's called the Nobel Peace Prize now, but and what those are is different types of, whether it's discoveries or anything that leads to the advancement of mankind. What the author would not go into with these two individuals. Her name is Barbara McClintock, and this would be back in the 1950s. That as far as discovering DNA, a little bit of background information is technically she was the one that really discovered this, but the information was, if you want to say, perhaps stolen from her lab. So just think your life's work, okay? Everything that's possibly putting you onto the track for the Nobel Prize is taken right from underneath your feet. And I can't imagine how, let's say, terrible that would be just to have that happen to an individual. But nonetheless, the history book was written. These two had actually discovered it. And these individual letters, the A, the T, the C, and the G are called individual what? It's up on here if we don't know. Yeah, they're individual nucleotides, okay? So a nucleic acid then, which we just drew, as an example, would be a long chain of repeating units, because some of those nucleotides do repeat with one another, okay, in the form of DNA, okay? Now, that green dashed line that you saw, what do you think that is up on here? Yeah, that's hydrogen bonds that holds the DNA molecules together. Now, that's going to be important for as we go forward, as you know, adenine bonds to thymine, cytosine bonds to guanine. We're going to talk about replication here and why that's important. Okay? So we did this on Friday as well. Does that sound right? Okay. So these, you're still writing? Okay. All right. Glad to see no one's trying to pull a fast one in here. Some students like to write their grades in pencil, so then after I sign it, they can change that. So if you're wondering why we gotta do grade reports, that's why. But you ladies, I don't think you have to worry about that because I think Mrs. Preheim takes care of that for you. Yes. Yep. Okay. So now, not quite yet. Soaking all this in. Oh, okay. And some people can. Well, I guess we're going to find out then. See if you can remember all this information, and maybe you can. I don't know. You don't. You try. Okay. Because as we get further into this concept here, I'm going to open up the biochemistry book. Doesn't that make you sound smart? You can just say, hey, we learned from a biochem book today. So then when you go home and talk to your parents, they're talking to you, and how was school today, and what's your answer? Just fine, okay. And for some people, that's the answer they want to give. Nothing wrong with that. Did you learn anything today? Was that ever asked? So what's your answer? Oh, see, that's a good answer. That's right. You know, lo and behold, I never really had heard this phrase before, and it applied up here too. For those of you who don't know, I did put a year of science in at O'Gorman High School. I just remember the gentleman next to me was the department head, and some of the students he had in his anatomy and physiology class, he just looked. I was talking to him one day, says, geez, I just can't believe it. I can see why our boys are one in the state, because they are so smart in class. He was talking about football and basketball. You know, it's okay to be smart and be athletic at the same time. You know, you don't have to think, well, geez, I don't want to look like I'm smart. I'm an athlete. Well, chances are, if you're smart in here, you're probably going to be making better decisions there and there. That's just an opinion, but I thought that was rather true. And then when we look at last year, I'm sorry, I got a mini Malik sitting here, right? That and Bodie, I think you'd be surprised on how smart he is as well. But that's neither here nor there, but moving on to this concept here, okay? we're going to start talking about these different steps. It starts here, these first two steps, and goes on to these two, okay? I just got to make sure to keep an eye on the clock, because that bus isn't going to wait, you know. Doesn't wait for anybody, probably not even for me, and I don't want to go running after it. So. Is that what I look like? Probably. I don't know. All right. Now, it's more than just those two gentlemen I had mentioned that are quite intelligent. Don't think I'm just limiting just to those two. Now, something looks like it got cut off on the bottom. Let me take a peek here. Transcription. I don't see it either. Okay, let me maybe just make this a little bit bigger. And hopefully that makes a difference. No, it does not. Okay. Come on, don't make me look stupid here. As if I could fail at that. Of course I could. I'll shrink this guy. And take your advice. See if that will do it. Aha. Genius. Whoever said that. Well, you could have took credit for that. How do I know you don't have a voice-changing apparatus in your pharynx? Or no, larynx, sorry. I could be mean, and maybe you gentlemen think, gosh, I wish sometimes the women would get laryngitis. Do we even know what laryngitis is? Does anyone know what your larynx is? Yeah, so what is that, though? This, for gentlemen, yes. Before I explain, I'm going to say my apologies, ladies. I didn't mean anything by that. Because your larynx is your voice box. If that's inflamed, well, then it means you can't do what? You can't talk. So that's why I said I'd throw that out there now. My apologies if I offended anyone. Pardon me. Excuse me. So, with this, one of the things you're going to have to wrap your head around this, DNA does not leave the nucleus. Because one of the things that we had said before is, okay, what's the center of the cell, and also called the command center? It's what? The nucleus. And this is why. All the instructions for that cell are found inside that nucleus. Whether it's an epithelial cell, a muscle cell, a liver cell, any type of muscle cell, I think I said skin already, or any type of neuron, nephron, any one of those different types of cells are going to have different functions, okay? So you would not want to have the same type of genetic information for an epithelial cell, which is the skin, inside one of your neurons. It just would not match up, okay? Because they have two totally different functions, okay? That's why we say in transcription, okay, DNA acts as the template, because DNA is where the instructions are found for that cell. How does that message get out to where it needs to be? Well, anytime you are sitting watching our gentlemen out there in the huddle on offense, you have a go-between who typically is going from the, if you want to say after the play ends, so from the huddle to the sideline, back to the huddle. Who's usually doing that? Okay, so we'll say it's Nate then, and the command center, okay, Who's telling you what to do? Okay, whether it's Mr. Williamson making a choice on whether to go for it on 4th down, or whether it's Coach Mertz giving you offensive plays, or even in volleyball. Do you have one of these where, because I haven't noticed it, where if I have other teams, the coach is standing here with a clipboard and they're doing one of these or one of these or one of these? No? Okay. Why would a volleyball coach do that? Okay? So, in other words, information from the bench is getting out to you on the court. It's the same type of concept here, to where, again, this is the coach that's making the decision. So this information has to get out to the ribosome, and it does that in the form of messenger RNA. So a better way of looking at that is Nate, because that's an actual individual rather than a number, okay? So I don't want to get too far into this, but we will make sense of this later down the road. But we want to talk about this DNA replication. So does everyone have this information that wants it? So what we will do here is this. We're going to take this double helix, okay, because it's got this structure on both sides. So that's why it's called a double helix. And what we will do is just picture, okay, this tongue depressor in this dimension. What we are going to do, okay, is unwrap that and turn it on its side. So we're taking this and turning it on its side and opening it up. But if you would look on, oh, we're having fun. What's he doing there? Does that say mount up or HUD? That's not your huddle call, is it? But there's that. Yeah. I'll stop there. Let's see here. We want you to look on page, going the wrong way. Around page 197. I said Barbara McClintock earlier, didn't I? This is in the textbook. Rosalind Franklin was her name, not Barbara McClintock. Okay. I knew it was a female that had probably discovered DNA. Okay. So with what you see on page 201, sorry. 201. Notice what you see there in figure 10-10, okay? You see one double helix there of DNA, okay? So again, what we are going to do is take this model, flip it on its side, and open it up. So what that means is we are taking it and doing this. I think this is a better way of looking at it, okay? So now, all of these red nucleotides we'll go ahead and put on the top, okay? So TTC, TTC, then GGC. Okay. I bet I'm going to run out of room here, aren't I? Okay, so now I think hopefully that's fairly accurate. Okay, so we said A, G, C, C, G, A, T, T, A, C, T, C, and then A. All right. So what we see happening there in figure 10-10, and what you had wrote down in your paper, okay, what we see happening is, notice we have those hydrogen bonds, okay, that we're tearing them apart. We still have those hydrogen bonds down here yet, because it's what enzyme that breaks them apart or breaks them bonds apart. This is just an enzyme moving in that direction. And what they're showing that as in this diagram 1010 is that purple circle that's moving up that replication fork. What is that called? DNA what? Not polymerase, that's too far down the line. Are you asking me or telling me? No. Because you said it. I thought. Helicase, yes. DNA helicase. So that is the enzyme that is doing this. I know you can just say helicase, but I'd rather have you know it as DNA helicase. So what is that function of that helicase enzyme, do you think? Whether you look at it in this diagram or what you wrote down in what we were talking about 10 minutes ago. What does that DNA helicase enzyme do? It starts to break something. But what's holding that DNA strand together? Maybe it didn't say it on there. I thought it did. Okay, DNA helicase is an enzyme that will split the hydrogen bonds. Okay, so remember those green dotted lines are hydrogen bonds. So this helicase breaks those bonds apart. Okay, then what we see happening is as these are broken, these bonds, okay, there's nucleotides that are floating freely in the nucleus. What happens is they start to find their natural base pairings, okay? Whether it's adenine with thymine, cytosine with guanine, okay? So you tell me, which would you rather work on, the top or the bottom? Okay? So we'll go ahead quickly. Okay, so what bonds with thymine? What bonds with thymine? Do we see, we wouldn't even have to go any further. Do you see a pattern here? It's color-coded for a reason. Okay, so what bonds with cytosine? Guanine, thymine, A, T. Okay, what do you notice about the outside of this fork here? This is the exact same that you see on the bottom here. Okay, so it wouldn't take a genius to figure out is if we start putting these complementary base pairings in, what's it going to be? The same as the inside top. The outside here is the same as the inside bottom. So the outside down here will be the same as the inside top. Is anybody just totally lost here? Okay, because it's just like what happens when you reach a fork in the road. This is a replication fork, but if you reach a fork in the road, what should you do? Pick it up. It's a fork. Pick it up. I told a funny, I see lots of disgruntled looks out there, or trying to break that wall down, and fork, pick it up. Maybe you got a salad you want to consume. Now you got your fork. But if it's got 3 tines, well then it's to trident. Maybe or look out. Maybe Neptune's not too far away. It's gonna strike and throw a lightning bolt at you. No, that's Zeus. I'm getting my Greek gods mixed up. Yeah. Okay. So what we see happening then, again, with thymine bonds, or excuse me, with adenine bonds thymine, okay, and then, shoot, I'm sorry, I don't got the black lines in like it did along the top, but I think you get the idea, okay? I'm not even looking what they're matching up with, I'm just copying what's on the inside. Providing you do that correctly, you would be able to do that. That's a G, C, T, A, T, G, A, C, oops, that's a G, and then T. Okay? I didn't even look at hardly any of these blue letters. I just copied the red ones. Does it look correct? Okay? So now, our 4 steps that we see happening. 1, DNA helicase is breaking these hydrogen bonds, or you could say it's kind of like a zipper that's unzipping that double helix, that double-stranded molecule. Then we got nucleotides that are just kind of hanging around, floating freely inside the nucleus, start to match up with their base pairings, that adenine bonds with thymine, cytosine bonds with guanine, okay? Then, what do you suppose is going to happen after this? Like in diagram 1010 or figure 1010, there's going to be a zipper that seals that strand back up again. And it's not DNA helicase, but here you have to be specific. It's DNA what? Polymerase, okay? So what happens is that's another enzyme, that's all that circle is, DNA polymerase. The reason you need to be specific with that is there's also RNA polymerase. That's a totally different process. Okay? So we're probably not quite going to get as far as what I had wanted, okay? But that's okay, because this is too important of a concept to just kind of skip over. I was hoping we might get a little further. And one of the things you'll notice, count up just these nucleotides on top. How many you get? Really? Is it 14? I was hoping it was one more. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Shoot, you're right. There's supposed to be 15. Okay? That's not your mistake. That's mine. Because this will become more important, I want you to look at page 207. On page 207, what do you notice about this table? All of these codons, it says codons in messenger RNA. We haven't talked about messenger RNA, but that will be one of the aspects we talk about tomorrow. But each one of these codons has how many letters in it? Okay? So when we do some of these DNA molecules, whether it is 18, 12, or 9, what would those 3 numbers have in common? 18, 12, 9, 15, 6. They're all factors of what? Do you think? They're all factors of 3. That's why we like doing these in 3-letter base pairings. I thought I had 15 on here. I kind of drew that up at the last moment. But, and we didn't, and I lied to you, Ryan. But we opened it, and we closed it. So we did open a biochem book. We didn't quite get as far as what we wanted. So with that, we will pick this information up tomorrow. Okay? And probably have your books ready. Come to class and have those books ready. If you don't have your vocab terms done yet, get them done by Friday. We'll have a vocab quiz then on Friday. Okay? Now, if this information seems like it's kind of daunting or overwhelming, this is one of them you really don't want to try to play catch up with. Okay? So know what those steps are in replication. DNA helicase breaks the hydrogen bonds. You got these nucleotides floating freely. They match up with their base pairings in step 3, and then step 4, DNA polymerase seals the strand back together. So again, we'll pick up with that tomorrow.